Welcome back, friends. This is Dr. Mlelwa Lecturers. Don't forget to click the subscribe button and to click the bell, then put all for more updates. To join our Telegram group for discussions, just click show more in the description. Then if you scroll down, you see the links for the O-level and the A-level group. Friends, this is coordination session 25, and in this session we are going to discuss about the synaptic transmission. In the previous session we discussed about the structure of the synapse, and as I said that we discussed the structure of the chemical synapse. So even in the process of synaptic transmission we'll be discussing about the chemical synapse, because in electrical synapse actually we don't have the process of synaptic transmission is how it occurs in the chemical synapse. So in the following account, the example of neurotransmitter acetylcholine will be used. Kwa hiyo tutatumia acetylcholine kama ni neurotransmitter ya kuelezea synaptic transmission. Kwa hiyo synaptic transmission inaanza kuelezewa pale ambapo nerve impulse inakuwa imefika kwenye synaptic knob. That means nerve impulse imefika at the end of the axon, ambao tulidiscuss in the previous session about the structure of synapse, na tukasema kuhusu bulbous terminale au the synaptic knob, yani the expansion at the end of the axon. Yani at the end of the axon ina divide into branches, then at the end of the branches we have the expansions called the synaptic knob. So when a nerve impulse explanation yake ya synaptic transmission inaanza when a nerve impulse arrives at the synaptic knob. What happens? It depolarizes the presynaptic membrane. Kwa sababu manake nerve impulse kama tunavyojua kwamba inapofika mahali fulani ina depolarize, inafanya depolarization. So the presynaptic membrane inakuwa depolarized. Depolarization ya presynaptic membrane inasababisha calcium channel is open. So we have the calcium channel in the presynaptic membrane, and when the calcium channel they open, they increase the permeability to presynaptic membrane to calcium. So opening ya calcium channel manake ita allow calcium ion kufanyaje kuingia. So opening of the calcium channel will allow the calcium ions to enter. If the calcium ions rush into the synaptic knob, they cause the synaptic vessels to fuse with the presynaptic membrane and discharge the neurotransmitter substance into the synaptic cleft. That is called exocytosis. Kwa hiyo the process of exocytosis inatokea hapa ambapo tunakuja kuona kwamba the synaptic vessels zina fuse with the membrane, lakini kinachosababisha synaptic vessels they fuse with the membrane is the entry of calcium ions. Kwa hiyo lazima uelewe hiyo simple mechanism. Cause ikisha fuse with the membrane they empty, manake zinavo fuse with the membrane then wana release ile neurotransmitter kwenda kwenye nini, kwenye synaptic cleft. Kwa hiyo manake tayari zile synaptic vesicles zinakuwa nini, empty. Zikishakuwa empty zinarudi kwenye synaptic knob. So the empty vesicles move back into the cytoplasm where all are later filled with the transmitter substance, that is acetylcholine. Kwa empty vesicles zinarudi kwenye cytoplasm, na zikirudi baadaye zitakuwa filled na nani, na acetylcholine. So we shall see later how the vesicles they are filled again with the acetylcholine and where the acetylcholine comes from again. Kwa sababu tulisema kwamba the neurotransmitter substance most of times they are proteins and they are made up in the cell body. So zikishatengenezwa kwenye cell body, zikishaenda kwenye synaptic knob, it is not all the time kwamba zinatoka kwenye cell body. Most of time kama after the nerve impulse transmission this acetylcholine is recycled. Sio kwamba ikishatumika kutengeneza kusafirisha nerve impulse ndio inapotea. So acetylcholine diffuses across the synaptic cleft, a process which takes about 0.5 millisecond. So kama tulivyosema kule mwanzoni, one among the difference between the electrical and the chemical synapse is the speed of conduction. That is, in the chemical synapse we have a delay of about 0.5 millisecond, Kwa nini kuna hii delay? Kwa sababu zile chemicals zinakuwa zina diffuse kwenye synaptic cleft, while in the electrical synapse you don't have this delay because the ions they diffuse directly from the presynaptic from the synaptic knob into the postsynaptic knob and attaches. So after they have diffused across the synaptic cleft, they attach to the specific receptor site, or sometimes they are called as protein molecule, or sometimes you may call them as receptor molecules on the postsynaptic membrane that recognizes the molecular structure of acetylcholine molecule. So the protein molecules, they have been modified kama tulivyosoma kule kwenye biochemistry kwamba every enzyme has its active site. So even most of the receptors, they work as how the enzymes they are working. So most of the receptors, they have the active site, and the active site is the one which recognize the exact structure of the molecule. Kwa manake kwa mfano hapa receptor site ambayo ina recognize acetylcholine, pale hawezi akaja sodium, hawezi akaja potassium, hawezi akaja nani, ni lazima aje acetylcholine. So receptor site inakuwa inajua kabisa the structure of acetylcholine is like this, and when the acetylcholine has already binded to the receptor site, then the level of the acetylcholine causes a change in the shape of the receptor site, which is at in the ion channel opening up in the postsynaptic membrane, allowing sodium ions to enter. So acetylcholine akishafika kwenye receptor molecule, akibind pale, the sodium channel zina open. Sodium channel ziki open, sodium zinaingia, na sodium ziki enter manake depolarization iliyokaa, and the normal process of the nerve impulse transmission itaendelea kama kawaida. Note that in the transmission of nerve impulse along the axon, ion channels are open in response to depolarization, whereas in the postsynaptic membrane of the synapse, it is in response to binding of the neurotransmitter to the receptor proteins. Kwa kama tulivyosema kwenye mechanism ya development ya impulse, jinsi gani ambavyo impulse inatengenezwa, and if you remember we said that the generation of the impulse is due to slightly depolarization caused by the stimulus, lakini kwenye postsynaptic membrane depolarization au opening ya sodium gates inatokea baada ya nini? Binding ya acetylcholine. So maana yake ni kwamba zile receptor site zenyewe ndio kama channel zenyewe, so zikibind to acetylcholine zina open, zile receptor molecules ina allow sodium to enter. So the entry of sodium ions through the postsynaptic membrane causes depolarization of the membrane. This excites the cell, making it more likely to set up a nerve impulse, that is action potential. Lakini sometimes if the amount of the acetylcholine released is not much, kinachotengenezwa sometimes cannot be an action potential in such, but can be a generator potential. However, it is not obvious to produce a generator potential, because most of times if the receptor has already generated the action potential, the size of the action potential does not change if it moves from one neuron to another. But sometimes due to some of the factors which we study later, some of the discussions on the summation, then we will understand that we can generate something or potential called a generator potential, and then a generator potential, if they combine many of them, or we can generate the excitatory postsynaptic potential, and then if they combine most of them, they make an action potential at the postsynaptic membrane. But the easier concept which you should understand here that after the binding of the neurotransmitter to the receptor sites, they allow the opening of the sodium gates, and then the sodium ions they rush inside the axon, then the depolarization occur, and it is most likely the nerve will produce the new action potential, and it will propagate the nerve in the nerve impulse in the second neuron. So once acetylcholine has depolarized the postsynaptic neuron, it is hydrolyzed by the enzyme acetylcholinesterase. So we have this enzyme, as I told you before, that you will know how the vesicles they are filled again with acetylcholine. Pale mwanzoni nikwambia kwamba tutakuja kuona huko mbele ni kivipi ambavyo vesicles zinakuwa filled with acetylcholine. Kwa hiyo acetylcholine mpya inatokea huku. Acetylcholine ikishafanya opening ya sodium gates, ikishafanya depolarization, inakuwa hydrolyzed, yani inafanyia digestion. So the acetylcholinesterase, sometimes called cholinesterase, its short term is A, capital C, then H, capital E. So acetylcholinesterase, which is found in the postsynaptic membrane. So this break down the acetylcholine to prevent breakdown of the acetylcholine is important because it prevents the successive impulse merging at the synapse. That means acetylcholine, if it has already been transmitted the impulse to the postsynaptic membrane, it should be digested, then it should be brought back to the presynaptic membrane. Kule inatumia energy kwenye presynaptic membrane, zina combine, so they combine acetylcholine and choline, they form acetylcholine again. Then that acetylcholine is packed again into vesicles, and at that point it is easier for the acetylcholine in the vesicles to transmit again the nerve impulse. Kwa maana if the acetylcholine ingekuwa hii broken down, ingekuwa narudi vile vile, sometimes impulse ambayo ingekuwa imetoka kwenye neuron ya nyuma ikija inaweza combine tena na acetylcholine zile, then the excitation of the postsynaptic membrane ingekuwa iendana na the amount of the acetylcholine released at the presynaptic membrane. So the resulting choline and ethanoic acid, that is acetyl, acetyl, diffuse across the synaptic cleft and are actively transported into the synaptic knob of the presynaptic neuron into which they diffuse. So the acetyl, that is ethanoic acid, and choline, they diffuse back through the synaptic cleft, and then they enter into the synaptic knob. So here they are coupled together again and stored inside the synaptic vessel, ready for further use. The combination of the choline and the acetyl requires energy, and the energy for coupling acetyl and choline comes from mitochondria, which are present in the presynaptic knob, which are present in the synaptic knob. So this coupling requires energy, which is provided by the numerous mitochondria found in the synaptic knob. Some nerve gases and insecticides work by inhibiting acetylcholine. So when we are talking of nerve gases, hizi ni gesi ambazo ukizivuta zinaenda ku break down the transmission of the nerve impulse, and then at the end of the day unajikuta tu ni kwamba nerve impulse inakuwa haiwezi kusafiri, and then the human being dies. So we have some of the few points to note here. The first thing is that some neurotransmitters are inhibitory in their effects. In some of the aspects it is a neurotransmitter, and in some of the aspects it is the neuron. So the inhibitory neurotransmitters haziendi kuinduce depolarization kwenye postsynaptic membrane, so they cause hyperpolarization rather than depolarization of the postsynaptic membrane. So hyperpolarization makes the inside of the cell even more negative, making it less likely to set up a nerve impulse. So inside of the cell ina become more negative, and in this case more especially it is because of the opening of the potassium channels instead of the sodium channels, if I'm not mistaken, and we will study this later. One among the neurotransmitter which is inhibitory is GABA, and we will study GABA later and some details of the other neurotransmitters. Also, there are 3 possible ways to remove neurotransmitters from the synaptic cleft. Actually, these are not ways, but just like stages, because we may reabsorb by the presynaptic membrane, we may diffusion out of the cleft, and hydrolysis by the enzyme. Actually, these are not ways, because the neurotransmitters, for example acetylcholine, should be hydrolyzed first, then it should diffuse into the presynaptic membrane, but sometimes they can diffuse out of the cleft, and if they diffuse out of the cleft, it means there will be the need to produce the new neurotransmitters in the cell body, so that they will transmit the nerve impulse in the next nerve impulse, which will pass through that neuron. So in this lecture, let us end up here, and in the next lecture we'll be discussing about the detailed mechanism of the synaptic transmission. In the next lecture we'll be discussing the synaptic transmission by stages, and how the process they are taking place by diagrams. So thank you everybody. Let me wish you nice studies.